Receptors for smell, olfaction, and taste, gustation, are chemoreceptors. They respond to chemicals in an aqueous solution, and they complement each other and respond to different classes of chemicals. Smell receptors are excited by airborne chemicals that dissolve in fluids coating the olfactory epithelium of the nasal mucosa, whereas taste receptors are excited by food chemicals dissolved in saliva.
Most of the nasal cavity is lined with respiratory epithelium, however there is a small patch of olfactory epithelium located on the superior part of the nasal cavity, specifically the superior nasal concha and the superior part of the nasal septum. Air entering the nasal cavity turns sharply as it enters the external nares, nostrils, and swirls around the turbinate bones, inferior and middle nasal conchae, to reach the olfactory epithelium. Because of the location of the olfactory epithelium, humans must draw air forcibly into the nasal cavity as in sniffing to intensify the sense of smell. The olfactory epithelium consists of olfactory receptors, supporting cells, and basal cells. Interestingly, the olfactory receptor cells only live for about a month and then must be replaced. The basal cells produce new olfactory receptors. There are 10 to 100 million olfactory receptors. The olfactory receptors are bipolar cells, as are many of the receptors for special senses, and possess non-modal olfactory cilia which greatly increase surface area for reception of olfactory stimuli. The olfactory cilia project into a layer of mucus produced by the supporting cells and olfactory glands in the underlying CT. The mucus is a solvent that dissolves airborne odorants so that they can bind to receptor proteins in the olfactory cilia membranes. This opens cation channels and creates a receptor potential which is carried by a short dendrite to the axon. Groups of unmyelinated axons of the olfactory receptor cells form fascicles known as the fila olfactoria or cranial nerve eye. The fila olfactoria project through the olfactory foramina in the cribriform plate of the ethmoid where they synapse with a second-order neuron called a mitral cell located in the olfactory bulbs. The mitral cells form structures called glomeruli and axons from neurons bearing the same kind of receptor converge on a given type of glomerulus. Each glomerulus represents a single aspect of an odor, but each odor activates a unique set of glomeruli. From the mitral cells, impulses flow from the olfactory bulbs in the olfactory tracts to two main destinations. The first pathway travels via the thalamus to the piriform lobe of the olfactory cortex and part of the frontal lobe just above the orbit, where smells are consciously interpreted and identified. Each olfactory cortical neuron receives input from up to 100 receptors for analysis. The second pathway bypasses the thalamus and goes to the hypothalamus, amygdala and other regions of the limbic system, which elicits emotion responses to odors. Smells associated with danger, smoke, gas, skunk scent trigger the sympathetic fight versus flight response. Appetizing odors cause increased salivation and stimulate the digestive tract, whereas unpleasant odors trigger protective reflexes such as sneezing, choking or gagging. Taste and olfaction have a lot in common. The sensory neurons of both are chemoreceptors. In the case of olfaction, odoriferous molecules must be inhaled and must dissolve in mucus coating the surface of the upper part of the nasal cavity. For taste receptors to work, materials must be placed in the oral cavity and must be dissolved in saliva in order to effectively stimulate the taste buds. The sense of olfaction is not well developed in humans but is 50 times more sensitive than the sense of taste. Olfaction augments taste sensations. Sometimes strong olfactants can override the sense of tasting what is in the mouth. Taste buds are the sensory receptors for the special sense of taste. There are about 10,000 taste buds located on various types of papillae on the surface of the tongue. There are 3 types of papillae on the tongue. The circumvallate or just valate papillae are the largest and least numberous with 7 to 12 forming an inverted V at the back of the tongue. The uniform papillae are muscromust and harbor the vast majority of taste buds. The fungiform papillae are scattered over the entire surface of the tongue. Foliate papillae are found on the side of the tongue. The third type of papilla does not contain taste buds. Filiform papillae are used for manipulating food when chewing or licking. It's these type of papillae that make a cat's tongue feel so rough. In addition, a few taste buds are also found of the soft palate, larynx and pharynx. The picture on the left is a diagram of a circumvallate papilla. The picture on the right is an enlarged view of a single taste bud. Each taste bud consists of 3 types of cells: the gustatory or taste receptor cells, which number about 50 per taste bud, supporting cells which form a capsule for the receptors, and basal cells. The basal cells replace old receptor cells which only have a lifespan of about 10 days because of the huge amount of friction they are subjected to on a daily basis. The taste receptor cells have long microvilli called gustatory hairs that project from the cells through a taste pore to the surface of the epithelium, where they are bathed in saliva. Sensory dendrites coil around the taste cells. They represent the first part of the gustatory pathway to the brain. Our taste sensations are complicated mixtures of qualities. When taste is tested with pure chemical compounds, taste sensations can all be grouped into one of 5 basic qualities: sweet, sour, salty, bitter, and umami. Sweet taste is elicited by sugars, saccharin, alcohols, and some amino acids. Sour taste is produced by acids, specifically their hydrogen ions in solution. Salty taste is produced by metal ions such as sodium chloride. Bitter taste is elicited by alkaloids such as quinine, nicotine, caffeine, and some non-alkaloids such as aspirin. Umami is a relatively new taste sensation discovered by Japanese scientists. It is elicited by the amino acids glutamate and aspartate, which appear to be responsible for the beef taste of steak, the tang of aging cheese, and the flavor of MSG. Taste maps assign the basic qualities to specific areas of the tongue. For example, sweet sensation was localized to the tip of the tongue, salty and sour to the sides, and bitter to the back. These maps have been proven to be false. Recent evidence shows that all modalities of taste can be elicited from all areas that contain taste buds. 3 cranial nerves are involved with carrying taste information. Cranial nerve 7, facial, serves the anterior 2/3 of the tongue. Cranial nerve 9, glossopharyngeal, serves the posterior 1/3 of the tongue, and cranial nerve X, vagus, serves the throat and epiglottis. When taste buds and their fibers are excited, these nerves relay this information to the sensory neurons in the solitary nucleus of the medulla oblongata. From there, taste fibers project to the limbic system, hypothalamus, and of course the thalamus. The thalamus relays taste information to the primary gustatory cortex located in the insula of the parietal lobe of the cerebral cortex.